Very good afternoon to all of you. Today we are here to discuss another topic that is metabolic pathway, which include central metabolic pathway, that is glycolysis, TCA cycle, fatty acid oxidation, triglyceride biosynthesis, and its catabolism. So it is quite lengthy topic, and I got the assignment to complete this lengthy topic in one lecture. So I will try to conclude. Although I hope these central metabolic pathway has been the part of syllabi since your +2 means +2 and graduation level of classes. So you all might be aware about these pathways. So wherever I will be getting the opportunity that, and I realize that you people know, I will try to go little faster. Okay. So so what is metabolism? So metabolism basically is the set of biochemical reactions, set of biochemical processes, which is very vital for survival of living organism. Okay, so these are life sustaining chemical processes, and basically these processes are required in order to derive the energy from the food molecules, food what we eat, and convert these food to the building blocks of body like proteins and necessary biosynthesis of lipids, nucleic acids, DNA, carbohydrates, and other, and even storage polysaccharides and other storage materials. And apart from that, the elimination of waste is also one of the crucial part of metabolism. So these, when we consider these metabolic processes for within an organism, okay, so then this is called as metabolism. Okay, then this is called as metabolism. And when we consider this metabolism within a cell, within a confined cell, then it is called as intermediary metabolism. So what we see, there is a difference in intermediary metabolism and this cell. This one, intermediary metabolism is a subset of metabolism where we consider the life sustaining chemical processes specific to confined within a cell, whereas in metabolism it is a wider concept where we consider all the processes, chemical processes within a cell and as well as outside the cell. Basically transportation of cells, transportations of metabolites across the cells are also considered in metabolism. So there are several classifications for the same, like anabolism and catabolism. It is a very basic concept. I hope you might be aware that anabolism is the biosynthetic process, all the processes relating to the biosynthesis of proteins, lipids, nucleic acids, carbohydrates, and other things, which is very essential for survival of living organism. So these are the part of anabolism, and the catabolic processes mainly you will be like the processes which are involved in oxidation of foods and the other nutrients in the food is the catabolic processes. Many time even the life sustaining molecules, which is part of the structural part of the body, can get also be metabolized or catabolized specifically to derive energy. Okay, and those structural part again will be replaced. So you might have heard about protein turnover. So in protein turnover in all our proteins, those are metabolic catalytic activity or structural proteins. They can also be they can also be digested or catabolized to derive this energy for the use of cells. And after a due time, those proteins, whenever cells require, that can be again synthesized. So these are the anabolism and catabolic processes And these things you might have heard, means you might be knowing. There is one more basic difference in anabolism, where you will see that most of the anabolic processes, they are reductive in nature. Reductive in nature means they will be, they will be processed under a reductive environment. Okay. For your recognition, I can quote that wherever those are the compartments, those are the cellular compartments where a reductive environment will be created. Like you have chloroplast. So in chloroplast, you have NADPH. Okay, so NADPH is a reducing NADPH, and NADPH to NADP plus ratio is very high in chloroplast and such biosynthetic compartments. So anabolism is always processed in a always done in a confined compartment where reductive environment is there, and these processes are reductive in nature. Whereas catabolic processes, these processes are oxidative in nature, and these oxidation releases energy, and which is we preserve those energy into ATP, and ATP is the energy currency of cell. Okay, and all the cellular processes will be needing those energy currency. So basically, these metabolic processes, these metabolic reactions we considers. So there are several reactions when there is a interconnect of the 2 or 3 or subsequent metabolic processes. Those processes, when substrate of one biochemical reactions or metabolic reaction is the product of previous metabolic reactions, or alternatively you can say the product of one previous metabolic reaction is the substrate for another metabolic reactions, and these sequence of metabolic reactions leads to a final products which has vital importance for the survival of living organisms. So these kind of processes are called these kind of interconnected metabolic reactions are called as metabolic pathway, and the number of metabolic reactions is unlimited in a pathway. So theoretically, hypothetically, you can consider a number of reactions in metabolic pathway, and further these metabolic pathway, for our convenience, we can distinguish it into either linear pathway, branch pathway, circular pathway, and into a number category based on our needs. So basically, linear, circular, and branch pathway, those kind of pathway, they have their own significance. Okay, everywhere you will be not finding why TCA cycle is a linear, why TCA cycle is not a linear pathway, why it is a circular pathway. So circularity of a pathway is defined by when the final product of a pathway is the substrate for the pathway. Okay, so because then it is called a circularity. Okay, when linear pathway is there is one substrate which is completely different from the products. Okay, so product cannot again can enter into the linear pathway, but in circular pathway you have the product which is also a substrate. Okay, so circularity of the process has several advantages. One of the advantages, all the intermediates will be consumed within the circle only. Okay, and siphoning of the intermediate from the circular pathway is basically it is difficult. You can feed the pathway, but siphoning of it requires several stringent regulations. So circular pathway are more suited for catabolic process, whereas linear pathway are more suited for this one, more suited for anabolic pathway. So similarly, you can there are several database after advent of bioinformatics. We have several of databases. Two databases, Metasys and Cake Pathway. These are two important pathway databases where you can find out details of each metabolic pathways. Okay, it has several curated metabolic pathway, around 2937 pathways from different organisms, and there are several kind of metabolic pathways also, which is very unique to a living organism. Like there are generalized pathway, or we can say there is a common pathway which is redundant across the organism. Okay, like you have glycolysis, you have Krebs cycle, you have electron transport chain, you have fatty acid metabolism, either synthesis or catabolism. These are the pathway you will find across the organism. You will find the pathway, but there are certain pathway which is very specific to organism. So those are called as specialized pathway. And you have several example, like we are dealing with the plant, and plant are known to have several specialized pathway, particularly of environmental or ecological significance. So like you might have heard about phenol biosynthesis, you might have heard about anthocyanin biosynthesis, you might have heard about several other terpenoids and terpene biosynthesis. So these secondary metabolism are also known as specialized metabolism. Am I audible? Am I audible? Please confirm.
Yes, sir.
Okay. So, coming to the our topic of significance, that is glycolysis. So glycolysis, why glycolysis? So basically glycolysis is nothing but the oxidation of glucose into pyruvate. Okay, pyruvate. So pyruvate is the intermediate metabolites which can be fed to the downstream pathway in the catabolic process. So glucose is basic carbohydrate, carbohydrate or sugars, which is the end product of digestion. Okay, so not only sugar but all the hexoses like glucose, lactose, mannose, these are the dextroses. So these are the end product of digestion. So after digestion, these glucoses in the cell, it get oxidized to pyruvate, and further this pyruvate in anaerobic conditions, it either get fermented to ethanol, or it can get reduced to lactate. Okay, so these are under anaerobic condition, and if aerobic conditions persist, then it can enter into tricarboxylic acid cycle, and where it will be directly oxidized, completely oxidized to CO2 and H2O. These are also oxidation. But these are the partial oxidation of glucose. Okay, but this TCA cycle leads to the complete oxidation of CO2 and H2O. Okay, here carbon is oxidized, carbon of glucose get oxidized, and hydrogen of glucose get reduced. Okay, oxygen of glucose get reduced. Okay, so glycolysis, basically we categorized into three steps, three stages. So the initial stages, what is done, that the sugars are attached with the phosphate moieties. So phosphate moieties are attached with these sugar molecules. So for attaching these phosphate moieties, we have different enzymes. Okay, we have hexokinase. So hexokinase can add phosphate group to different kind of hexoses. Okay, so again hexokinase is of different kind and with different km constant, different substrate specificity. So these hexokinase, what it does, it phosphorylates these glucose molecules, and we get a glucose 6 phosphate. Why does this phosphorylation takes place? Okay, why this glucose 6 phosphate, this phosphorylation takes place? Actually, you can see, you will be knowing that you might not be knowing, but phosphorylation phosphate is a negatively charged group. Okay, so when phosphate is attached to a glucose molecule, so what happens? It cannot be transported across the membrane. Okay, it cannot be transported across the membrane when it is a negatively charged. Why? Because membrane, the outer portion of the membrane is a hydrophilic. Okay, outer portion of the membrane is hydrophilic. Okay, so hydrophilic, and the core is the hydrophobic in nature. So transporting those negatively charged molecule across the membrane, you require a lot of energy. Okay, so by phosphorylating those glucose molecule, you can confine the sugar molecule into a compartment. Okay, so what it does, when you phosphorylate, means all the molecule will be within a confined environment. So there is a different kind of enzymes which can access those metabolites. Okay, so the chances of reactivity enhances several folds. So in the first step, hexokinase phosphorylates this glucose into glucose 6 phosphate. Further, this glucose 6 phosphate get isomerized in presence of PGI, phosphoglucose isomerase, into fructose 6 phosphate, and this fructose 6 phosphate further phosphorylated by phosphofructokinase into fructose 1,6 bisphosphate. We also call FBP. Okay, 1,6-BP. So these three steps is called as investment step, where we are attaching, we are adding, or we are spending ATP molecule for phosphorylation of these sugars. Okay. Similarly, if you have different other than glucose molecules, then those glucose molecule can also be phosphorylated. Those sugar molecule can also be phosphorylated by hexokinase. Even it is not phosphorylated to hexokinase, there are other kinases which can phosphorylate it, and if it is not, then it can be converted to directly to fructose 6 phosphate. Okay, fructose 6 phosphate. So there are isomerases which can convert galactose kinase into galactose 6 phosphate into fructose 6 phosphate. So similarly, such type of enzymes are there in the cellular machinery. So in the initial stage, what we get, so one glucose molecule get transformed into fructose 1,6 bisphosphate. So this is called the investment, or investment of ATP, where glucose is phosphorylated. And second, basically this first stage, what it does, it imparts negative charges and concentrates the glucose into the confined environment. Okay, in the second stage, what it does, this 6 carbon fructose 1,6 bisphosphate get hydrolyzed into 2-3 carbon intermediate. First, and both can be interisomerized, like dihydroxyacetone phosphate can be isomerized into glyceraldehyde 3 phosphate, and the vice versa. Okay, so again, after this glyceraldehyde 3 phosphate has the potential to enter into the downstream of this pathway. So glyceraldehyde 3 phosphate will again get into the third stage of the reaction, and since the glyceraldehyde 3 phosphate is entering into the reaction, so automatically this reaction equilibrium shifts toward glyceraldehyde 3 phosphate, and this sets the flux for downstream glycolysis process. Okay, so what happens, so glyceraldehyde 3 phosphate it get dehydrogenated in presence of dehydrogenase enzyme into 1,3-bis-phosphoglycerate. So where here you have since dehydrogenases, so you generate one NADH per glyceraldehyde 3-phosphate molecule. So similarly, you have later this glycerate molecule get further one phosphate molecule will be released and 3-phosphoglycerate will be formed, and these 3-phosphoglycerate will be mutated to 2-phosphoglycerate, and further action of enolase and pyruvate kinase will lead to the production of pyruvate. Okay, so pyruvate is the final product of glycolysis. So this has several effects we discussed in the earlier slide. So if you go through the initial like each step, so initially what we do, so glucose molecule in presence of hexokinase, we transfer phosphate moiety and glucose 6-phosphate is produced. So these kinases, it has hexokinase has different specificity for glucoses under different biochemical conditions. Okay, so same hexokinases, if present in liver, it behaves differently, and when same hexokinase is present in blood, RBC cell, so it behaves differently. In what respect it behaves differently? So it catalyzes the same reaction, but the substrate specificity and the kinetics of the reaction get changed. So similarly, phosphoglucose isomerase and the second reaction where glucose 6-phosphate get isomerized into fructose 6-phosphate, and further this fructose 6-phosphate get converted into fructose 1,6-bisphosphate in presence of phosphofructose kinase. You will see. So this is the in earlier, these are the, you see, these are the reversible reactions. Okay, so this is the irreversible reaction. Hexokinase is irreversible reaction. Okay, so irreversible reactions always, in most of the cases you will be finding that irreversible reactions requires certain amount of energy or driving force. Okay, and since it is irreversible, it is very strictly regulated. Okay, similarly, second this phosphofructokinase is also irreversible reaction, and it is under strict regulation of strict regulation of ATP to ADP ratio and Mg2 plus. Okay, so here what it does, fructose 6-phosphate get here, one phosphate group is here, and again one phosphate will be attached here, so 1,6-bisphosphate will be formed, fructose 1,6-bisphosphate will form. So this is very rate limiting step for photosynthesis. So phosphofructose kinase is the rate limiting steps of glycolysis. So basically, PFK reaction is the first unique and irreversible step in glycolytic pathway, and it is rate limiting, and it is the first pathway which commits the sugar molecule, phosphorylated sugar molecule, into the glycolysis. Okay, so before, why this is, because you see we have several pathways where you will be phosphorylating sugar moieties. Okay, even in biosynthetic pathways also, even in biosynthetic process also, you will be phosphorylating these sugar molecules. Okay, so there also, if it is phosphorylated, it doesn't mean that it will be entering into glycolysis process. Okay, so when it will be confirmed, when this phosphofructose kinase, then this phosphofructose kinase will double phosphorylate, means another phosphate group will be added. So this is, since it is irreversible reactions and ATP is consumed, so this reaction sets the flux for the glycolysis pathway. So second stage is catalyzed by one enzyme called aldolases. So aldolases we have discussed in enzymology class. So this fructose 1,6-bisphosphate get in presence of aldolases, it get not hydrolyzed, it get split into dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. So aldolases are the class of enzyme which is not involving any water molecule. Okay. So dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. So what you will see DHAP, we call it DHAP and glyceraldehyde 3 phosphate. So DHAP, this dihydroxyacetone phosphate, it has several utilities. Okay, even glyceraldehyde 3 phosphate also have several effects in biochemical, like metabolism. Okay, several effects. So glyceraldehyde 3 phosphate is further, in presence of dehydrogenase, will be set into the further glycolytic process. Okay, and you will be like during fatty acid biosynthesis, you will be reading about how glycerol or glyceride is synthesized. Okay, and even how the reductive pentose phosphate pathway siphons off this dihydroxyacetone phosphate from this glycolytic pathway. So whenever required cellular process, whenever required any intermediate is required, these intermediates are being pulled off. Okay, for the vital processes in cellular processes, not only energy. So energy is the main objective, but whenever other functions of those metabolites are needed, so those metabolites will be siphoned off from the pathway. Okay, although definitely siphoning off has cost, so that will be done. Okay, so further, this dihydroxyacetone phosphate and glyceraldehyde 3 phosphate, they are under equilibrium and in presence of triphosphate isomerase. So when you are using this glyceraldehyde 3 phosphate in the downstream pathway, automatically this equilibrium will be shifting toward the forward reaction. Okay, so all since one glucose molecule will be producing two molecules of dihydroxyacetone phosphate, so those two molecules will be converted into glyceraldehyde 3 phosphate, two molecule of glyceraldehyde 3 phosphate. So further, these two molecule of glyceraldehyde 3 phosphate will be attached with another inorganic phosphate in presence of NADH, and this glyceraldehyde 3 phosphate dehydrogenase, what it does, it hydrogenates this NAD plus, and NADH is produced. So this NADH is the reducing equivalent produced in the glycolytic pathway, which will be oxidized in the oxidative phosphorylation process in mitochondria. Okay, so this is the glyceraldehyde 3 phosphate. So this is the first reducing equivalent formed in the glycolytic process. So, and further, this 1,3-bis phosphoglycerate takes another form, another ADP, and directly undergo a substrate level phosphorylation, and this ADP is converted into ATP. Okay, so this is the first ATP produced. Okay, so phosphoglycerate kinase is the first enzyme which yields substrate level phosphorylation in in glycolysis process. Okay, similarly, in then each steps has some unique this one. So if you consider one glucose molecule, so there will be 1,3-bis phosphoglycerate, so two molecule of ATP will be produced. Okay, so if you see the like this is the phase 1 where we are spending energy. Okay, one is here and one is here. 2 ATP per glucose molecule is spent for phosphorylation of glucose and this splitting. Okay, further, in the second energy generation process, so in energy generation process, first we get two molecule of NADH here, then two molecule of ATP here, and in the final you have pyruvate kinase, which convert this phosphoenol pyruvate into this pyruvate molecules. So here also you will be producing two molecule of ATP. You remember, wherever irreversible reactions are there, so these reactions are endergonic in nature. Okay, you cannot reverse the reactions. Okay, you cannot reverse the reaction. Otherwise, gluconeogenesis and glycolysis both are similar pathway running in opposite direction. But these are the places where irreversible reactions are there, and these irreversible reactions cannot be reversed. Okay, so there they have different enzymes involved to have a bypass of those reactions. Okay, so these things we have discussed. So if you will see this glucose to pyruvate conversion, so you have in glycolysis, you have how much ATP in first stage, you 2 ATP is consumed, okay, and you have 2 NADH and 2 FTH. Okay, so in the first stage, what we have seen that you consume 2 ATP, okay. This glucose is phosphorylated with the help of 2 ATP, and one glucose molecule generates 2 molecule of this glyceraldehyde 3 phosphate. So here onward you will have 2 coefficient. So here 2 ATP is produced, here 2 NADH is produced, and here 2 ATP is produced. So total of 4 ATP is produced, of which 2 is consumed earlier and 2 NADH is produced. So this 2 NADH has energy equivalent of 3 ATP. Like 1 NADH is equivalent to 2.5 or 3 ATP. So this will be considered as 6 ATP. Okay, so 6 ATP, so that you can consider. Many times you will see that these NADH is considered as either 2.5 or somewhere 3 ATP. So there is a difference in considering that 3 ATP and 2.5 ATP. So that we will discuss when we will discuss chemiosmotic process. Okay, so under anaerobic condition, what it does, so under anaerobic condition, why we do glycolysis? So basic principle, basic foundation of performing glycolysis is that any living organism require energy, right? Any living organism require energy. So if in a linear metabolic pathway, if any substrate is accumulated, any product is accumulated, okay, any product is accumulated, so accumulation of products gives a feedback inhibition, okay, feedback inhibition to the pathway, and that feedback inhibition will stop the metabolic reactions, okay, that will block the metabolic process. So what happens, or even acute shortage of any metabolites also or co-substrate also can block the process. So what happens if there is an anaerobic condition? So in anaerobic condition, what it does, so this pyruvate in presence of NAD plus, okay, NAD plus, it generates NADH and CO2 and H2. Okay, this is in the oxidative condition. Okay, in anaerobic condition, what it does, whatever NADH is produced in the glycolysis pathway, so that NADH is consumed here. Okay, NADH is consumed here to reduce this pyruvate into lactate. Okay, to reduce this pyruvate into lactate. Similarly, under fermentation also, this NADH is consumed to produce this ethanol. This is fermentation. So what happens if it is NADH is not consumed? Okay, if NADH is not consumed, then what will happen? If NADH is not consumed, then this NADH will create a reducing environment. Okay, reducing environment is a reductive equivalent. Okay, reducing equivalent. So if it is more, if you go on, you just doing the linear pathway of glycolysis, you will have plenty of NADH molecule. Okay, because NADH is produced here, NADH is produced here. Okay, so if you will go on performing this reaction, so you will have plenty of NADH molecule. So once this amount of NADP plus this NADH is more, okay, once this just a minute, once this NADH, okay, once this NADH to NADH to NAD plus, okay, this NAD plus Okay, NAD plus ratio goes up. Okay, up, it hinders the catabolic process, because catabolic process is a oxidative process. Okay, catabolic process is a oxidative process. So under reducing environmental condition, reducing cellular condition, this catabolic process cannot be done. And this is the basic principle, because this is the basic foundation because of which we are having different cellular organelles with specific functions. Okay, in mitochondria we have catabolic processes are going on in mitochondria, so that is why we call powerhouse of cell. Okay, so this NADH is reducing equivalent. So to utilize this NADH, these fermentation or reduction process happens, homolytic fermentation or alcoholic fermentation happens, and by this fermentation this NADH is consumed, and at least the 2 ATP whatever produced here. Okay, whatever produced here, 2 ATP and 2 ATP, 4 ATP produced, out of which 2 is consumed, so these 2 will be lost, so only 2 ATP will be produced, because this NADH whatever it is produced here, it will be consumed in case of anaerobic condition. Okay, so this this reduction is basically important for consumption of NADH. Okay, so basically this ethanol is how ethanol is produced. So pyruvate get decarboxylated. Okay, pyruvate decarboxylase, so that produces acetaldehyde. Okay, that produces acetaldehyde, and acetaldehyde further reduced, okay, dehydrogenated, reduced to an ethanol. Okay, how it is reduced? This is the carbonyl group. This carbonyl group is reduced to hydroxyl group. Okay, so this happens. So similarly, when you have plenty of you have other sugars, like if you have fructose, so fructose directly it can fructose 1 phosphate by fructokinase, it can convert into fructose 1 phosphate, and this fructose 1 phosphate can be easily either means easily split into glyceraldehyde and dihydroxyestone phosphate. So further this glyceraldehyde can be acted upon by glyceraldehyde kinase, and it will be glyceraldehyde 3 phosphate will be produced. So this glyceraldehyde 3 phosphate can enter easily into glycolysis pathway. Okay, so fructose, this is how fructose enters into glycolysis. So similarly, if you have galactose, so galactose can be easily converted into galactose kinase. Okay, galactose kinase. Okay, and galactose kinase can be isomerized to glucose phosphate. Okay, sorry. This galactose can be in presence of galactose kinase, it can be easily converted into galactose 6 phosphate, and galactose 6 phosphate can be isomerized to glucose 6 phosphate. Okay, so glucose 6 phosphate can further be isomerized to fructose 6 phosphate, and fructose 6 phosphate can directly enter into the glycolysis process, where we have fructose 1,6 bisphosphate formed. Okay, phosphofructose kinase actions will convert this bisphosphorylation. Okay, so after this one, again all the reactions of glycolysis will happen. So in summary, if you see in all, either you have trihaloges in insect, or lactoses, or sucroses, or even glycogen starch, all can easily be at various places can enter into glycolysis process. Okay, so at glucose 1 phosphate you have galactose which can easily enter into glucose 1 at glucose 1 phosphate. Okay, similarly you have glucose, so which can enter at glucose 6 phosphate, and you have these fructose molecule, so which can either directly get hexokinase, in presence of hexokinase can enter here in form of fructose 6 phosphate, or it can alternatively be acted upon by fructose kinase and phosphorylated at first positions, and further it aldolases produces glyceraldehyde and dihydroxyestone phosphate, and glyceraldehyde first will be acted upon by kinase and form this fructose glyceraldehyde kinase, and those can be directly enter into the glycolysis. So when like anaerobic, this we have discussed. So coming to the opposite of the glycolysis, we have gluconeogenesis. It is one of the metabolic process where we produce glucose uh, from non-carbohydrate precursors. Okay, from non-carbohydrate precursors. So why we will be doing gluconeogenesis? So reason is, like many times when cells are in a starving condition, okay, when cells are in a starving conditions, so in that conditions glucose is required. Okay, when you are in fasting, so brain's glucose level is dropped very fast. Okay, so in that conditions body produces glucose from non-glucose, non-carbohydrate moieties, non-carbohydrate carbons, basically present in various amino acids, even in fatty acids. So that carbon skeletons is utilized for production of those sugars, and the needs of the sugars within a cell or the within organism is met. Okay, so this is gluconeogenesis process. So what we do in gluconeogenesis process? So basically every all the process of gluconeogenesis will be will be entering at pyruvate level. Okay, so whatever the intermediates are being cooled off for production of glucose, so that will be converted into pyruvate, and after pyruvate, this pyruvate will be converted into glucose. How it will be converted into glucose? So all the processes whatever we have discussed till now of glycolysis, so opposite of that process is catalyzed within the cell, and that process is called as gluconeogenesis. Okay, so pathway, but both are same. Okay, both are same, but both are not performed at the same place. Okay, gluconeogenesis you will be performing in gluconeogenesis you will be performing cytosol, or even in plant you will be performing it in chloroplast, but in all the glycolysis process you will be performing it, that also is performed in cytosol, but later part portion of the glycolysis, like oxidation of pyruvate, will be performed into mitochondria. Okay, so the pathway from phosphoenol pyruvate to glucose 6 phosphate is common in the biosynthetic conversion of any different precursors to carbohydrate. So this pathway is called as gluconeogenesis pathway. Okay, so what we do in gluconeogenesis pathway, both are same. You can see in the opposite direction. First, what we do in the gluconeogenesis, this pyruvate is carboxylases. Okay, in presence of pyruvate carboxylase, what we do, we convert it into pyruvate oxaloacetate. Okay, pyruvate carboxylase, what it does, it carboxylates the pyruvate and convert into 4 carbon oxaloacetate. Okay, or this oxaloacetate can be directly coming from the TCA cycle. Okay, TCA cycle intermediates, either you take alpha ketoglutarate acid, or you can take succinate also, so or you can take malate also. So those intermediates can be directly converted to oxaloacetate. So these so when it is converted into oxaloacetate, so oxaloacetate can directly enter into gluconeogenesis process, whereas when it is from other amino acids, it can be directly it can be converted into pyruvate, and pyruvate will enter into gluconeogenesis process. When pyruvate enter into gluconeogenesis process, you have one active ATP is consumed here. Okay, one ATP is consumed here, so you have energy cost when it enters. Pyruvate enters into the gluconeogenesis process. Okay, so the action of pyruvate carboxylase sets the flux for gluconeogenesis process. It is a irreversible reaction. Okay, pyruvate carboxylase. So further in this auxiliary state, it will be another specific unique enzyme of gluconeogenesis process is the phosphoenol pyruvate carboxykinase. Okay, carboxykinase means it is carboxylated pyruvate. Okay, it is carboxylated pyruvate. Auxiliary state is what? Auxiliary state is nothing but carboxylated pyruvate. Okay, so this carboxylated pyruvate is phosphorylated, and that phosphorylated product is phosphoenol pyruvate. So you remember that in glycolysis, what was the last product? Pyruvate. The penultimate product was the phosphoenol pyruvate. So converting this pyruvate into phosphoenol pyruvate, we have two specific enzymes in gluconeogenesis, that is pyruvate carboxylase and phosphoenol pyruvate carboxykinase. Okay, so another these three reactions, that is enolase or phosphoglycerate mutase, phosphoglycerate kinase, and glyceraldehyde 3 phosphate dehydrogenase, these are the common enzymes we have in the process. Okay, we have in the process of gluconeogenesis and glycolysis, where these action of these four enzymes converts this phosphoenol pyruvate into glyceraldehyde 3 phosphate. Okay, glyceraldehyde 3 phosphate. So these glyceraldehyde 3 phosphate with the action of aldolase. Okay, with the action of aldolase also catalyzes reversible reactions. Okay, aldolase is basically what it does, it is kind of condensation reactions it performs, and these aldolases uh, that condense this 2-dihydroxyacetone phosphate and glyceraldehyde 3 phosphate into fructose 1,6 bis phosphate, and this phosphorylated sugar is formed. Okay, phosphorylated sugar is formed. So once you have phosphorylated hexose sugar, so what we have, you see, you remember that we had phosphofructokinase. Phosphorylation of this fructose 6 phosphate into fructose 1,6 phosphate in case of glycolysis. But here, to carry out this opposite reaction, you have a unique enzyme called fructose 1,6 bisphosphatase. Okay, bisphosphatase. So what it does, it removes phosphate from the first place. Okay, it removes phosphate from the first place, and removes first place, and you have fructose 6 phosphate. Okay, so again this fructose 6 phosphate will be isomerized into glucose 6 phosphate, and again glucose 6 phosphate, with the action of glucose 6 phosphatase, you will convert this glucose into glucose 6 phosphate into glucose. So you just remember, if you know the process of glycolysis, if you know the process of glycolysis, if you remember the process of glycolysis, just reverse of the process of glycolysis is the gluconeogenesis process with 4 unique enzymes. Okay, with 4 unique enzymes, you have pyruvate carboxylase, and that also you can easily remember if you know the irreversible reactions here. There you had HK, okay, hexokinase. So in case of HK, you have here glucose 6 phosphatase. Okay, in glycolysis you have PFK, phosphofructose kinase. Okay, you have phosphofructose kinase. So instead of phosphofructose kinase, you have fructose 1,6 bisphosphatase, and in the third here, the last, where you have phosphoenol pyruvate kinase. Okay, there you have one kinase, pyruvate kinase you have. So there in glycolysis you have pyruvate kinase. Okay, so pyruvate kinase. So in glycolysis you have pyruvate kinase, and here in gluconeogenesis you have two unique set of enzyme. Basically these enzymes are membrane bound, and you have phosphoenol pyruvate and phosphoenol pyruvate carboxykinase and pyruvate carboxylase. Okay, so carboxylase is basically what is, it is a carboxylation reaction where we are adding uh, carbon dioxide to a intermediate. Okay, so gluconeogenesis, basically these 4 unique set of enzyme what we discussed. So here you see glycolysis and gluconeogenesis, both are similar processes, only you have these processes where you have unique set of enzymes involved. Okay, in, and that is different in gluconeogenesis and glycolysis. So here you have pep carboxylase and pyruvate carboxylase, and your fructose bisphosphatase and phosphatase. Here you have hexokinase, phosphofructose kinase 1, and pyruvate kinase. So if you consider the energy equivalent, since glucose is biosynthesized, so always biosynthesis is an endergonic process, and there you will require energy. Okay. So for in terms of energy, if you consider, if you convert this 2 pyruvate molecule, which is a 3 carbon compound, so the 3 carbon compound into a 6 carbon compound, which is a glucose. So then you will be using 4 ATP and 2 GTP molecule. Okay, 4 ATP and 2 GTP molecule. Along with that, 2 NADH molecule will be utilized, and your glucose will be produced. So for producing each glucose, you will be using 6 ATP, 6 ATP equivalent. Okay, 6 ATP equivalent and 2 NADH. Okay, 2 NADH. So this also you can easily remember. If you know the reaction, you can easily remember. So here you see, here also you have utilized, here also utilized, you see ATP, here also it is utilized, here also ATP is utilized. So if you're converting 2 pyruvate, so 2 ATP will be utilized here. So NADH, reverse of catalyzing reaction, so this NADH will be consumed. So 2 NADH will be required here. So no other requirement is there. So this ATP consumption can also be memorized. Just spare me 1 minute. Just I'm putting my laptop in charge. Am I audible?
Hello, sir, you are audible, sir.
Yes, yes.
Yes.
So again, in pentose phosphate pathway, this is third pathway. This is biosynthetic pathway, another biosynthetic pathway. So what we, as name suggests, pentose phosphate pathway. Here we also oxidation, oxidation, and that is performed only in specific cells. Okay, specific cells it is performed, and alternatively it produces oxidation process is not complete oxidation. Okay, pentose phosphate pathway, you will have the glucose will be oxidized, but complete oxidation will not be done. So we have this is also called hexose monophosphate pathway. Hexose monophosphate pathway. So what we do here glucose 6 phosphate. Glucose is converted into glucose 6 phosphate, will be transformed into different lower carbon sugars like ribose or 4 carbon or 5 carbon sugars, and this oxidative phase will produce NADPH. Okay, this oxidative phase will produce NADPH, and these NADPH helps helps to maintain that reductive environment in a biosynthetic compartment. Okay, so these hexose monophosphate pathway or this pentose phosphate pathway is performed in the cellular compartment where biosynthetic processes are going on, and although it is oxidation of glucose, but the major objective is not to derive energy, but to get these reducing equivalent out of the oxidation of glucose. Okay, why? Because in a cellular process, in a biosynthetic compartment, there is a reductive environment has to be maintained. Okay, now how a reductive environment will be maintained? So this NADPH will be formed there. So NADPH to NADP plus ratio will be higher in those compartments. Okay, so in order to maintain that ratio, this pathway is being carried out. Okay, so you see these NADPH, either this will be acted by glutathione reductase. So this reducing equivalent will be generated. This reducing equivalent will be used for reductive dehydrogenation processes. Reductive dehydrogenation processes in the cellular biosynthesis. And the same can be various precursors can be reductively biosynthesized to various essential metabolites. So this is so initially here in the initial reaction only one CO2 molecule is released. Okay, so this is the only step where oxidation of this carbon to CO2 is done. So a no, there is another phase also where we convert this this glucose into 5 carbon sugars. So this is non-oxidative phases, and these non-oxidative phases are very common in photosynthetic cells, particularly plants. So in photosynthetic cells, in the dark reactions, you might have heard that where we have glycerol PGA, phosphoglyceric acid, and dihydroxyestone phosphate. So those through a chain of transketolases and transaldolases actions creates various sugar intermediates, and finally we get an pentose sugar that is ribulose phosphate, and that ribulose phosphate continues the photosynthetic process. So that non-oxidative phase is very crucial in generating very specific substrate for essential metabolism, metabolic reactions. Okay, so here you see in HMP pathway what is done here. So here this ribose 5-phosphate, this is 5 carbon, this one, this 5 carbon compound can be through series of action of transketolases and transaldolases, you have different carbon sugars, okay, and that will be formed into glucose phosphate, and that glucose will be producing entering into that oxidative mode of phosphorylation, oxidative mode of HMP pathway, and that oxidative mode of HMP pathway will generate enough reducing equivalent required for biosynthesis of various essential metabolites. Okay.
Hello. Hello, I'm audible.
Yes, sir.
Dr. Ashok sir.
Hello.
हाँ sir.
Yes,
हाँ, sir.
my voice is audible.
Yes. You are audible, sir.
Yeah, actually, there was connected internet connectivity
Yes,
issue.
sir.
Actually, I am using mobile phone, and I hope my video is off.
Oh,
My
yes,
video
yes,
is
yes,
off now.
yes. Your video is
This
also off.
will ensure
I
better
think there is a
connectivity.
poor bandwidth, I think so.
Yes, 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 sir.
Ah,
Actually, I am using that mobile phone. In previous days, I was from the home only. I was taking the lecture,
okay,
so there
okay.
is no issue. I have Wi-Fi there, but here in institute we do not have.
Okay, okay,
Okay,
okay, okay.
so
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 this is big
so
major
that is
issue.
yes, yes, yes.
So I think
Things are being worked out. Yes,
uh, okay, okay, okay.
things are being worked out here. Maybe in few days it will be here. So lipids are very important nutrients or
Sir, your slide is not visible, sir.
okay, okay, just a minute.
Please share your activity.
Just a minute, sir. Hello.
Ah, sir. Ah,
Yes,
yes, yes. It's
is it
visible.
visible?
Yes, yes, yes.
Yes, yes.
Visible and clearly audible.
Okay, so basically lipids are very important biomolecules, and it has been the part of very noticing components. Like you have various pigments in the eyes. Okay, cones and rod cells are there having different pigments. You have carotenes, various vitamins working as a cofactor, vitamin K, various detergents, very essential for digestion process. You have various transporters, very specific transporters anchored to the plasma membranes. So those are also having lipid moiety, dolichols. Is example. Several hormones are basically steroids, basically synthesized from various fatty acids, and various extracellular and intracellular messengers, icosanoids, phosphatidyl inositol derivatives, and various messengers are there of lipid origin, and various membrane proteins have these moieties for attachment into the membrane. So since the lipid has diverse role in the cell, so these lipids are on the continuous basis, it is being synthesized into the cellular process cell, and at the same time this lipid is acting as a storage biomolecules in most of the organisms. So those storage molecules need to be catabolized whenever energy needs are there for the cellular processes. Okay, so that is catabolism as well as synthesis is very essential for survival of this one. So basically, acetyl-CoA dehydrogenase is an mm, is one enzyme which is basically uh, involved in catabolism of this fatty acids. So that we will be seeing in this process. Okay, in this process we will be seeing this. So first we will look into the biosynthesis process, then we will go for catabolism. Okay, so basic monomer unit of this fatty acid biosynthesis is acetyl-CoA. So how this acetyl-CoA is formed? So acetyl-CoA is basically we have heard, we just gone through the glycolysis process. So in the glycolysis, what we did, so this glucose is converted into pyruvate. Okay, so this pyruvate further converted into one pyruvate molecule is further converted into acetyl-CoA molecule in presence of pyruvate decarboxylase enzyme. Okay, pyruvate decarboxylase enzyme. So this is a pyruvate decarboxylase enzyme is a very complex, like a multifunctional protein type of enzyme having several component. So that produces acetyl CoA, and that acetyl CoA is the precursor molecule for synthesis of these fatty acids. Okay, so basic, but these the two processes, both fatty acid catabolism and the fatty acid synthesis, both are happening in different locations. Okay, so fatty acid biosynthesis is basically, in the cells or tissues where the storage of these fatty acid is required. So those tissues will be having more of these vital organelles, and there the synthesis of fatty acid will be more. And in other contrary the catabolic processes, so basically most of the processes of catabolism where you have mitochondria and other organelles, there you have peroxisomes and all. So there you have catabolic process of this biosynthesis will takes place. So both catabolism of fatty acid and anabolism of fatty acid, both are either temporarily isolated, or they are isolated in different organelles. Okay, so even in fatty acid storage tissue also, both processes happens, but both processes happens at a different time. Okay, so that is one very crucial thing. So and because both the requirement of synthesis and catabolism is entirely different. So first step of this synthesis, first step of synthesis is the malonyl CoA synthesis, malonyl CoA synthesis. So how this malonyl CoA is synthesized? So basically this malonyl CoA is synthesized through acetyl CoA carboxylase. Okay, so acetyl CoA carboxylase, we abbreviate it as a ACCase. So this ACCase has three major, this is one of the multifunctional protein, so has several domains. So it has three unique protein components. One is biotin carboxylase, another is trans carboxylase, third is biotin carrier protein. Okay, those three components, trans carboxylase, biotin carrier, and biotin carboxylase, these are three functional domain of this ACCase enzyme. So basically biotin carrier protein, what it does, it has a biotin molecule. Okay, biotin is a precursor, biotin is a vitamins, you might be knowing. So this biotin molecule is attached with the biotin carrier protein. So this biotin molecule plays a very crucial role for this ACC carboxylase reactions. Okay, anywhere where you have transfer of that carbonyl group, so that transfer of carbonyl group is catalyzed in presence of biotin only. Okay, so here this BCP protein has biotin moieties. So what it does, this biotin carboxylase, what it does, so what it does here, you see this biotin carboxylase domain, it binds with the carbonate. Okay, carbonate ions will bind here, and after binding, this biotin will take this carbonyl. Okay, take this carbonyl, and it will transfer this carbonyl, this carbonate ion, this carbon from this biotin carboxylase domain to this trans carboxylase domain, and this at this trans carbylase domain, this carbonyl group will be transferred to this acetyl CoA molecules. Okay, so binding basically what is this is a simple condensation reactions. Okay, so acetyl CoA molecule, this whatever the you have this nucleophilic O minus, it will donate the electron here, and this condensation of acetyl CoA with carbonyl group, it will lead to malonyl CoA synthesis. Okay, so this malonyl CoA synthesis is the malonyl CoA is the precursor for biosynthesis, and it is being synthesized continuously with the help of ACCase. and since it is very crucial and very rate limiting, so this is tightly regulated. Okay, and many times you can see here this carbonate ion and ATP. This is ATP dependent reactions. So ATP will convert this molecule into carboxylated substrate. Okay, this is general reaction for any kind of this carboxylase type of enzymes. Okay, so this three domain structure, it is common in mammals, but you have these the similar proteins with multiple domains in different kind of different living organisms. Like you have in E. coli, you have 4 domain. Okay, like actinomyces, you have separate 2 subunits. Okay, alpha, beta, and even epsilon. And even tuberculosis also, you have different domains. So anyway, means my point is there is a lot of functional diversity in this ACCase enzyme, and because of which this enzyme has been targeted for designing various inhibitors being used in our herbicide industry. Okay, so what is this fatty acids? Apart from that, once malonyl CoA is synthesized, so another molecule, another protein, multifunctional protein, is the fatty acid synthase. So fatty synthesis is also a very big, large multimeric enzyme. So this multimeric enzyme also has several domains having specific catalytic properties, specific catalytic functions. So these functions are like unique enzymatic functions. Each domain has. So although the protein is of single, okay, multimeric single protein, but they have several domains having different catalytic functions. Okay, so here fatty acid synthase, you see you have 1, 2, 3, 4, 5, 6, and 7 units. Okay, so 7 units. So first one is beta ketoacyl ACP synthase. So beta ketoacyl ACP synthase, malonyl acetyl CoA ACP transferase MAT, and then you have dehydratase, then you have reductase, then you have ACP reductase, and then you have a carrier protein which can, like they have unique arm-like structures. So that arm-like structure helps them to bind the substrate and transfer the substrate from one catalytic domain to the other catalytic domain, and the final is the thioesterase, which will be releasing that finally synthesized substrate, finally synthesized product. So here you have, you see in the fatty acid metabolism, what individual steps, what is there, like malonyl acetyl transferase, what it does. So in this step, you have this malonyl is transferred to either acetyl or already existing any acetyl group. Maybe like already 6 carbons are there, so another 2 will be added like that. So this malonyl acetyl transferase will transfer that malonyl acetyl malonyl acetyl moiety, and then you have second enzyme where there is a condensation reaction. So both the already existing acyl group plus this malonyl group, you will get condensed, and you have this structure where you have 2 carbonyl groups separated with 1 CH2. Okay, so once it is there, so now this can be easily, get hydrated. So this hydration reaction will be carried out by this. Okay, so this can be easily reduced. So after reduction, this will be producing like alcohol. You see here, this will be reduced. So this CHOH, this C double bond one will be OH, and here H. Okay, so CHOH. So this is beta ketoacyl ACP reductase. it reduces this carbonyl group, and after reduction it produces this beta ketoacyl ACP, and then you have third enzyme dehydratase. So what dehydratase does? So it removes water molecule from there. So it removes water molecule there. So after removal of water molecule, you form a double bond. Okay, that E double bond will be formed. Okay, so after double bond, again it will be reduced. So again this will be reduced, and after reduction this will be the CH3, CH2, CH2. So this carbonyl group will be out with the reduction, okay, and one will be out with the this dehydration, okay. So this is fatty acid. So finally this chain will be elongated. So basically this method, these all four reaction is repetitively done in order to add another carbon group. Like you see here, 1 carbon, 2 carbon, 3 carbon, okay. So another CH2 if you wish to add, so you have to go through each 4 step again, okay. So again this C1 another malonyl CoA will come, and that MAT will transfer that malonyl CoA, okay. MAT will transfer that malonyl CoA to the enzyme and ACP enzyme. Then after transfer, both the substrate will come in the contact, and this condensation reaction will happen. And after condensation reaction, you will be getting this type of structure, means 2 carbonyl groups separated by CH2. And once this structure is formed, then this will be further reduced in presence of this beta ketoacyl ACP reductase. And after reduction, you will have OH, okay, OH and H. So this this can be water molecule can be removed from one H from here and one OH from here, so that you will be having a double bond structure here, and that dehydratase will create a double bond structure, and after double bond you can easily reduce that into with the help of reductase. And after reductase, this will be one CH2 will be added, and once the entire process is done, okay, means each cycle is done, so in the last you will be removing that that entire fatty acid with the help of thioesterase. Okay, so this is common process will be used for each like addition of carbon molecule in the fatty acid. Okay, like if you have to synthesize 16 carbon fatty acid, so 16 carbon fatty acid you need to have how many? 1 acetyl CoA and 7 malonyl CoA. Okay, so after so each time you will have to go through like acetyl CoA will be converted into propionyl CoA with all 4 steps. Okay, again propionyl CoA will be added by another carbon with subsequent 4 stages. So if you for synthesis of 16 carbon fatty acid, you need to go through 7 rounds of that entire process, 4 reactions. So that is why to have an efficient mechanism, we have a very complex multimeric enzyme, multifunctional protein having all domains, all catalytic enzymatic domain into one enzyme, and have a substrate channeling mechanism, which is of pantothenic acid. One prosthetic group is there, pantothenic acid, which binds with one substrate and transfer that substrate to other catalytic group, and from there to other catalytic group. So substrate channeling is done by pantothenic acid. Okay, so here also you have you see this kind of structure you have. So the basic here the mechanism is illustrated. So initially this acetyl group, first acetyl group will come, and here is the malonyl. So this malonyl acetyl transferase is the enzyme which will transfer this malonyl group to these enzymes. Okay, and then acetyl group is already here. So first step is condensation. So this will be condensed. So after condensation, you have alpha and beta, you see separated by 2 carbonyl group. Okay, so after once you have this, then you will be doing one reduction process. So reduction will be simply you will be having OH and H. So once it is reduced, then you have dehydratase. So dehydration will be removing this OH and H from here. So you have double bond formation. So once double bond is formed, this will be reduced with the help of NADPH and this one. So this is the basic basic mechanism of double bond formation and subsequent reduction. For forming double bond, you first have to reduce it. Okay, reduce the carbonyl group, and after reducing, you have to dehydratase action. Then dehydratase action will create double bond, and after double bond, you will be that will be acted by reduction, and that reduction will create another carbon group and addition of another CH2 group. So synthesis, if you consider the synthesis of malonyl CoA, 7-acetyl CoA needs 7 ATP for synthesis of 7 malonyl CoA. So 7 ATP is consumed. So similarly, 7 cycle of condensation is required for production of palmitic acid. Okay, so you have if you this is one assignment, you go and on the self you can calculate the amount of ATP. So for 16 carbon fatty acid, you will be requiring malonylation of 7-acetyl CoA. Okay, why not 8? 1 acetyl is of 2 carbon. So 7-acetyl CoA is malonylated, and first acetyl CoA itself is used. Okay, so 7-acetyl CoA, 7 ATP is required. Then further, this 7 cycle of synthesis will require 7 NADPH. Okay, so 1 NADPH you can consider 3 ATP or even 2.5 ATP equivalent. So based on that, if you consider for synthesis of this 16 carbon fatty acid, you will have 7 3 21 plus 7 ATP. Okay, means 28 ATP. But in the last thioesterase enzyme, action of thioesterase also it requires GTP. Okay, so there you will be another ATP will be consumed. So 27 ATP will be required for production of one fatty acid of 16 carbon. Okay. So further this palmitic acid with the action to, like in most of the mammals or in organisms, you have low chain fatty acid. Okay, low chain fatty acid, and both basically those are mostly saturated. Okay, saturated fatty acid. But in plants, you have plenty of non-saturated fatty acids. Okay, unsaturated fatty acid, and unsaturated fatty acids allow plants to have liquidity. You might have heard that when fatty acid is saturated, then it is solid at room temperature. Okay, so most of the oils which is liquid means they are unsaturated. Okay, and many times you might have seen that ghee, it is in solid form, means it has full of saturated fatty acid. So the most of the animals, mammals and all, they have full of saturated fatty acids, and plants have plenty, like very high amount of unsaturation in fatty acids, and basically it allows plant, it has advantage for the plant, and by which it can maintain fluidity in the membrane even at lower temperature. Okay, and it has several other advantages for fatty acid for plants, and if it harbors unsaturation, the catabolic process also become easier. Okay, when it is means harboring unsaturation in the chain, and the catabolism or reactivity of that fatty acid is much faster. So the turnover of fatty acid is much higher if it is unsaturated. So these are the several advantages of having unsaturation for the plants. So in plants, we have several unsaturated fatty acid of various chain length and various degree of unsaturation. So these unsaturations are means basically these means saturations are unsaturations are or desaturations are caused by mixed function oxidases and fatty acid desaturases we call, and basically palmitate will be desaturations will lead to palmitoleate, and further if you elongate it you will become 18 carbon saturated fatty acid stearate. Then further elongation it will lead to larger saturated fatty acid, or either if you will desaturate it it will become oleate, which is one unsaturation at 9, and further desaturation will lead to production of further alpha linoleic acid or gamma linoleic acid, and finally to omega 3 and omega 6 fatty acid pathway with the action of subsequent action of elongation and desaturation will lead to production of longer and higher desaturated fatty acids in the plants. Okay. So if you consider further is the once fatty acid is synthesized, so how this fatty acid is used in how this triglycerol is a part of most of the storage lipids. So how these lipids are synthesized from fatty acids? So this is a very common question. So this is glycerol 3-phosphate. Glycerol 3-phosphate glycerol is the part we were discussing, that we have dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Okay, glyceraldehyde 3-phosphate. So just reduction of glyceraldehyde 3-phosphate will lead to dephosphorylation, and reduction of glyceraldehyde will lead to glyceraldehyde or simply reduction of glyceraldehyde 3-phosphate will lead to for production of glycerol 3-phosphate. Okay, so glycerols directly this can be siphoned off from glycolysis glycolytic pathway. So this glycerol 3-phosphate with the action of fatty acid, this is varying chain of fatty acid. So acyl transferase enzyme will be there. So it will transfer one R1 to this OH group and another R1 to this OH group. So you have one phosphatidic acid. So in phosphatidic acid, you have two acylated carbon. Okay, two acylated carbon. Okay, fatty acylated carbon and one carbon attached with the phosphoric acid. Okay, so further if with the action of phosphatase it will produce diacyl glycerol. Okay, diacyl glycerol. Further acyl transferase action will lead to production of triacyl glycerol. So further these acyl transferases are these are generalized and specific kind of acyl transferases. So many times you have fatty acids of different natures, like higher long chain fatty acid, short chain fatty acid, saturated higher level saturated fatty acid, so monosaturated polyunsaturated fatty acid. So these acyl transferases reactions are many times very specific reactions required for synthesis of a specific lipid molecules. Okay, so if you if you go through the biomolecules fatty acid, then you will come across with various kind of lipid molecules, and what are the differences in those lipid molecules. Based on that you can look for the enzymes involved in the biosynthesis of that. So oxidation of fatty acid is further you can easily it can be easily visualized as, like, first we will go through the beta oxidation. So in beta oxidation what it does, so what are the beta bond we have created, beta the CH2. Okay, whatever beta CH2 we have created, so each will be broken by in this oxidation process, and this beta oxidation is basically the reverse of what we have used in the synthesis. Okay, so all the enzymes whatever we have used in the synthesis process, so same reverse action will be required for oxidation of that fatty acid. Okay, that process might be catalyzed by different enzyme, but my intention is that process is just reverse of that. Okay, so after beta oxidation you will be forming acetyl CoA will be produced, and that acetyl CoA can directly enter into TCA cycle. Okay, TCA cycle, and TCA cycle will produce reducing equivalent NADH and FADH2 and even ATP, and that FADH and NADH will give electrons or give electrons to for substrate this oxidative phosphorylation in mitochondrial electron transport chain, and finally ATP will be synthesized. So coming here also you have four enzymes involved in the oxidation. So you consider acetyl, first enzyme is acyl CoA dehydrogenase. So here one FADH2 is produced, and in this process you will be you will be producing this alpha and beta. This bond will be after dehydrogenation you will create one double bond. Okay, one double bond. Once double bond is formed, you will hydrate it. Okay, hydrate it. So there we were doing dehydration, so here we are doing hydration. Okay, after hydration you will form enol. Okay, you will form enol. You have the OH and OH. Okay, so this bond becomes very susceptible now, and after again you will be having an action of dehydrogenases, so it will remove this H and OH, so another carbonyl group is restored. So once carbonyl group is restored, another action of acyl CoA acetyl transferase thiolase will remove acetyl CoA. Okay, acetyl CoA will be removed, and one carbon less, two carbon lesser fatty acid will be left. Again, this will be going another round of beta oxidation for production of this. So in this process, you can see one H2O molecule is consumed, one NADH is produced. Okay, one NADH is produced, and one FADH2 is produced. Okay, so FADH2 is basically means each cycle you will be getting one FADH2, one NADH. Okay, one FADH2 and one NADH. So based on that, based on this, you can you can calculate how many beta oxidation cycle is required for conversion of this 16 carbon fatty acids. So for my calculation, what I have done for palmitoyl CoA is 16 carbon, so that 16 carbon will lead to like 7 round of beta oxidation. Okay, 7 round of beta oxidation will produce 7 round of beta oxidation will produce how much? Okay, sorry, 7 round of beta oxidation will create 7 FADH and 7 NADH. Okay, 7 FADH and 7 NADH. So FADH2 and NADH, FADH2 has 1.5 or 2 ATP equivalent, or NADH has 2.5 or 3 ATP equivalent. So based on that, you can calculate for 16 this one 16 for 16 ATP 16 carbon fatty acid, you will through beta oxidation only you can generate 7 FADH, means 7 to 1.7 to 214 ATP, and 7 NADH means 3 ATP if you assume, so 21 and this one 21 and 14, this is 35. Okay, 35, and further this acetyl CoA will be oxidized in the TCA cycle, and so if you go on calculating, it will generate 108 ATP per palmitic acid. Okay, one fatty acid oxidation on chain length will lead to production of 108 ATP. Okay, and overall holistically, if you see that this fatty acid, how this you can see this triglycerol or lipid. First it will fatty acid will be hydrolyzed, fatty acid will be removed, and after removal of this fatty acid you will be left with glycerol and fatty acid. So glycerol will directly enter into glycolytic pathway. with the help of glycerol kinase, and this fatty acid can enter into this beta oxidation. Okay, so beta oxidation, this will happen, this fatty acid will enter into beta oxidation. Okay, this fatty acid will be transferred into the mitochondria with the help of acyl carnitine translocase enzyme, and this translocase will transfer this fatty acyl enzyme from cytosol to mitochondria. Here in mitochondria, you have beta oxidation process. So this fatty acyl CoA, with the help of oxidation, hydration, reduction, and thyrolysis process, you will be completing this beta oxidation process of fatty acid. So each means each round you will be releasing once acetyl CoA. Okay, so for 16 carbon, you need to go around 7 round of cycle. Okay, so what we'll do today, we will conclude here, and tomorrow, while doing photosynthesis, we will take that Krebs cycle and electron transport chain along with that ATP synthesis and other theories of ATP synthesis there only. Okay. Hello, sir.
Yes, sir. Yes, sir. Yes, sir.
Over to you, sir.
Yes, sir. Yes, sir. So thank you, sir. You deliver very nice lecture, sir. And I hope this is also included in the syllabus. And,
Yes, yes, yes.
This is a good opportunity for the student. So
Yes, any queries? Actually, very few students are there.
Yes, yes,
Only
yes, sir.
6 I can see, including us. So anyway, any queries and
so if you have
any
any
queries,
queries,
Prateek, Suvarna,
Pratik,
Venkat,
Chandana, Suvarna, ma'am.
oh, Suvarna ma'am. Okay.
She is a faculty, I think. Suvarna Poddar, ma'am. She is a faculty.
Oh, okay, okay, okay. Sorry, sorry.
So
I
she
don't
also preparing for NET ARS examination. So she is continuously engaged in the lecture. Sincerely, she is attending
Okay, okay.
the lecture. Ma'am,
Actually,
you have any queries, ma'am?
Topic is so vast, and
Yes,
like
yes, sir.
this will it takes a course. These topics takes like entire semester. So concluding entire semester into 1 hour lecture is very
It's
difficult.
too difficult task, sir.
Yes, it is
Actually,
very
we are
difficult.
not concluding this session. Just means the remaining parts are there, and we will continue on tomorrow. That's it.
Yes, 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 yes.
So
anyway, sir, tomorrow we will meet.
Yes, sir. Yes, sir. Yes, sir. So
Okay.
is there no question? So you can ask on tomorrow also. Okay. So
Yes, yes, yes.
So thank you, sir. Thank you very much for delivering such a nice lecture, and hope so most of the student will understood properly this concept.
Okay, thank you, sir.
On behalf of the Postgraduate Institute, MPKV Rahuri, we are very much thankful to you for accepting the invitation and delivering lecture on this topic, sir. Thank you very much, sir.
Okay, thank you, sir. Thank you.
Okay, sir. Okay, see you.
So, sir, this is enough for today. Signing off.
Yes,
Thanks.
yes, sir. Please.